Dear students, in this module, I'm going to give you an example for each type of the domain architecture that we looked at in the previous module. So let's start by reviewing the different domains that exist. So foremost are the alpha domains that are mostly constituted by alpha helices. And then there are the beta domains which are constituted by beta sheets. And then alpha plus beta, or alpha over beta, or alpha and beta. As well as there are the membrane domains that exist on the surface of the different cells. So here in this figure is an example of the first type of the domain. So here is the alpha domain. So alpha domain is essentially a lot of alpha helices that have come together with the help of these loops. The loops are shown in green. I'm highlighting some of them for you. So if you have such a protein, then you can assume that it belongs to it has an alpha domain. So this protein is essentially hemoglobin, and here it is its ID. So you can simply classify this protein to be belonging to an alpha domain. So this protein has an alpha domain and it performs certain functions that are representative of alpha domains. So after looking at hemoglobin, I'll show you immunoglobin. So this is a different protein as compared to hemoglobin, and that it is constituted by beta domain. So here you can identify the beta sheets in yellow as shown here, and that these are linked up by the loops in red. So if you have such a protein, you can then classify it to have a beta domain in it. So in the first example, we looked at an alpha domain protein, and it had lots of alpha helices, or actually purely alpha helices. And in this case, this is a beta domain. So it contains purely beta sheets. Now we will move to a little more complex structure. So this is the alpha over beta domain. And as you can see that we have some alpha helices that are shown here. And right in the middle, we have some beta sheets. And then at its center is the alpha helix again. So let me clear this diagram for you and indicate the alpha helix at the center, followed by beta sheets surrounding it, and this is followed by alpha helices again. So if you have such an organization of secondary structures, then we call this alpha over beta domain. So essentially, the alpha helices are separated from beta sheets, and then alpha helices may come again, but they'll be separated and clearly at a distance from the beta sheets again. Next is the alpha plus beta. So in this case, we have the lysosome as an example, and you can see there are these beta sheets that are isolated at one side, and you have the alpha helices shown on the other side. So this is pretty simple to remember as well, that there is a clear separation of beta sheets and alpha helices. So this is different from alpha over beta in the previous slide, where they were mixed together in the architecture. So in conclusion, there are various types of domain architectures that exist, and each one of them can be classified by looking at the organization of secondary structures within the overall protein. Also, these structures can be classified into specific types, and databases can be built on which type of domains exist in which proteins. And these databases can be mined for further structural or evolutionary studies.